Hello, everyone. Welcome to Cancer in the Crosshairs. My name is Doug Kellogg, and I'm a member of the Department of Molecular Cell and Developmental Biology at UC Santa Cruz. I also do research on cancer, and I'll be the moderator for tonight's event. So tonight, we're going to hear about some of the exciting cutting-edge cancer research being carried out at UC Santa Cruz, all of which was funded by the Santa Cruz Cancer Benefit Group. So before going further, I want to emphasize that we are all here tonight because a little over 25 years ago, 5 friends saw a need in the community and made a promise over a glass of wine that they would work together to raise money for cancer patients and their families, and also for cancer research. These 5 founded the Santa Cruz Cancer Benefit Group, and since then, along with many more members, they have raised over $2 million to support cancer patients and cancer research right here in our community. So I want to take a moment here at the beginning to honor their selfless hard work and dedication to fighting cancer. If you've been to any of their fundraisers, you know that the glass of wine remains central to everything that they do. So I'd also encourage you to attend their next event, which is the Gourmet Grazing in the Green event on Saturday, October 8th. This is a wonderful event that combines food, wine, community, and fundraising for cancer. And I try to go every year, so don't miss it. The Santa Cruz Cancer Benefit Group has been supporting cancer research since 2005. So in doing that, they've helped researchers launch dozens of new cancer research projects. In addition, the Santa Cruz Cancer Benefit Group created an endowment at UC Santa Cruz to support talented undergraduate student researchers working on cancer. This support helps recruit and train the next generation of cancer researchers and oncologists, and you'll hear from one of these students tonight. So we're going to hear 3 short talks about cancer from 2 faculty researchers and 1 student, all of whom were recently funded by the Santa Cruz Cancer Benefit Group. I also want to emphasize that the support from the Cancer Benefit Group is very important. At UC Santa Cruz, more than 30 faculty are doing cancer-related research. Almost all these faculty have large research labs supported by grants from the National Institutes of Health and other major organizations like the American Cancer Society. Support from the Cancer Benefit Group has an outsized impact because NIH funding always comes with strings attached. It's awarded to fund very specific projects, and it's awarded in a very conservative way. So those of us who try to get funded from the NIH make the joke that to get a project funded by the NIH, you basically have to have it halfway done. And the reason is that the grants are awarded in a very conservative way because the funding groups want to see that a project is going to make progress and get data. And so they tend to be hesitant to fund riskier innovative projects. So that's where the Santa Cruz Cancer Benefit Group comes in. The $20,000 faculty grants from the benefit group help spark new cancer research by giving researchers the freedom and the power to start interesting new projects. And very importantly, the work funded by the Cancer Benefit Group can help lay the groundwork for getting a much larger grant. So for example, the Santa Cruz Cancer Benefit Group has awarded over $500,000 to faculty research projects since 2005. That $500,000 has laid the foundations for larger grants totaling more than $28 million. And it's led to progress in understanding and treating cancer. Some great examples of recent awardees emphasize this. So for example, Karen Ottoman, a faculty member, her project to enhance treatment of bacterial infections that cause gastric cancers had just received a $1.25 million grant from the National Institutes of Health. Chris Vollmer's project to identify better targets for cancer immunotherapy recently earned a $1.25 million grant from the National Institutes of Health. The American Cancer Society awarded Zhu Wang $750,000 to continue his work investigating how epigenetic regulators affect the progression of bladder cancer. And a great example is Daniel Kim's work to develop blood tests to identify cancer at the early stages. This is incredibly important because too often cancer is diagnosed too late to be fully treatable. His project was highlighted in Newsweek just a few days ago. All of these projects were funded by the Santa Cruz Cancer Benefit Group first. Again, emphasizing how these seed grants can help leverage much bigger grants and leverage big progress. Okay, so before introducing our most recent awardees for their talks, I want to give a very brief overview of cancer. What is it and why has it been so hard to cure? Cancer is personal for all of us because about 1/3 of us here will get cancer. All of us know someone who's had to fight cancer. My mother's a breast cancer survivor and I lost an uncle to cancer. And I have 2 close friends who are fighting metastatic cancers right now. Cancer is also personal because it's caused by mutations in our own genes. These mutations effectively create new kinds of cells that no longer obey the rules that dictate when and where cells should grow. Since cancer cells are mutant versions of our own cells, the challenge in cancer treatment is finding drugs that kill cancer cells without killing our normal cells, which is very difficult. Unfortunately, many cancers are still incurable. Current treatments extend life, but many patients ultimately succumb. So today's basic research at UC Santa Cruz lays the foundations for early diagnosis, improving current treatments, and discovering new drugs. Tonight, you'll hear from a chemist, Professor Michael Stone, and two molecular biologists, Professor Olena Vaske and undergraduate Sachin Parecadan. Michael is developing a novel cancer therapy. Olena is finding new ways to beat cancers in children, and Sachin is working in a faculty laboratory to better understand how and why cancers metastasize. And by the way, both Olena and Michael were deeply involved in our campus's developing our campus's COVID diagnostic laboratory, which was crucial for getting the campus through the COVID crisis. Okay, so I'm going to introduce our speakers now. After each one speaks, you'll have a chance to ask questions. Please put those questions in the Q&A, and then I will help the speakers see those questions and answer them. And our first speaker is Michael Stone. He's a professor in the Department of Chemistry and Biochemistry. And his lab has been focused on the molecular mechanisms that maintain the ends of chromosomes. This may seem esoteric, but it turns out that one way that cancer cells become immortal is by overriding these mechanisms. Tonight, he's going to tell us about an exciting and entirely new project aimed at developing new cancer therapies. So Michael, you can take it from there.
Okay, just need to unmute. Thank you so much, Doug, for that introduction. And thank you so much to everyone for being here tonight. I'm really excited to have an opportunity to spend a few minutes describing to you some of my team's recent work to develop novel antisense RNA cancer therapeutics. So many of you will be familiar with the principle of the central dogma of molecular biology. The central dogma describes the way in which information encoded in our DNA genomes is used as an instruction manual of sorts in order to make and maintain healthy cells and multicellular organisms. Now, in order for the information embedded in our DNA genomes to be useful, it has to first be decoded into an RNA molecule through a process called transcription. And many of these newly synthesized transcribed RNA molecules will go on to be decoded in a second step called translation to form a newly synthesized and unique protein molecule, which can fold into a three-dimensional shape in order to perform its unique cellular function. And proteins perform a wide diversity of cellular functions, ranging from maintaining the structural integrity of cells all the way to catalyzing critical biochemical reactions that are required for normal cell growth and maintenance. Now, our genes can generally be thought of as being organized into two types of sequence. There are exon sequences, and these are regions of our genes and RNAs that contain the necessary information to encode for a protein. And the other type of region is called an intron, and these are non-protein coding regions. So in this basic cartoon schematic, what I'm showing is the general principle of how a gene in our cells might be organized, where you have these information containing or protein coding exons interrupted by these non-coding introns. Now, in order for this RNA to become fully functional, what we call a mature RNA that can go on to be translated into a functional protein, these introns need to be precisely removed from the initial RNA, and the exons need to be joined together in just the right way so as to give rise to this mature functional RNA. And when this goes wrong, you can get non-functional RNAs and lose functions of certain proteins. So this process of removing introns and joining together the protein coding exons is referred to as RNA splicing and will be central to my talk this evening. So it's been appreciated for quite a long time that genetically inherited mutations in RNA molecules or in our genomes that ultimately are transcribed into RNA molecules are linked to human disease. And depending on the estimate, it's presumed that about 15 to 50% of genetically inherited diseases, and in particular cancers, arise from mutations that in one way or another affect the fidelity or the faithfulness of the RNA splicing process. I'm just listing here 4 different genes from our genome that my team and colleagues here at UC Santa Cruz are currently working on. The BRCA1 gene, patients that have mutations in this critical gene are at a significant predisposition to breast, ovarian, and prostate cancers. Some of the other genes we're working on are a gene called SMN1, which stands for survival of motor neurons. Mutations in the SMN gene give rise to a very severe degenerative disorder called spinal muscular atrophy. And more recently, we've started working on a few different genes, a gene called F8 or factor 8, which is a blood clotting factor. Mutations in F8 can give rise to type A hemophilia. And most recently, we've started working together with one of our other speakers tonight, Olena Vaske, together with her colleagues at UCSF on a gene called DEGS1. Pediatric patients that have mutations in DEGS1 can sometimes exhibit what's called hypomyelinating leukodystrophy, which is also a very debilitating and fatal pediatric neurodegenerative disorder. So excitingly, over the past several decades, a new type of RNA therapeutic has been developed, and that's called antisense oligonucleotide RNA drugs, or ASOs for short. And some of these ASOs can actually be designed to bind to a very specific target gene that might be carrying one of these genetically inherited disease-linked mutations, and can actually tip the balance from producing a non-functional RNA toward producing a functional RNA, and in doing so, correct this problem and treat certain diseases. The term antisense in ASO refers to the manner in which the drug interacts with its RNA target, obeying the simple logic of DNA and RNA base pairing, where you may be familiar with G's pairing with C's specifically, and T's pairing with A's. And so that's what gives ASO drugs their high degree of precision and specificity. So perhaps one of the most groundbreaking examples of an ASO RNA drug that's been approved by the FDA is a drug called Spinraza, which is used to treat spinal muscular atrophy, which I described earlier. This is a very high efficacy treatment, and really demonstrates the power and potential of ASO RNA drug development on the one hand. On the other hand, the several decades that it took to actually do the necessary groundwork to discover Spinraza and get it to a point of FDA approval, together with the rather high cost of administering this therapy to patients who need it on an annual basis, that those 2 points really highlight some of the existing challenges we as biomedical researchers face if we're interested in broadly applying the power of ASO RNA drugs to different disease contexts, for example, cancer. So recently, my team and colleagues at UC Santa Cruz have been working toward developing or identifying appropriate BRCA1 mutation carriers that might be candidates for the development of a precision ASO therapy. So just to tell you a little bit about what BRCA1 is a really important protein in our cells that helps to repair DNA that's been damaged, and it's also involved in the replication or the copying of DNA during cell division. BRCA1 is known as what's called a tumor suppressor gene. Tumor suppressor genes are often compared to the brake in your automobile in the sense that this type of gene helps to put a check on the amount of, or the speed with which a cell can grow and divide. And you can imagine that when a mutation occurs in a tumor suppressor gene that breaks this function, now, as Doug was describing earlier in his introductory remarks, a cell loses its ability to control its growth properties, and that is really a hallmark of all cancer subtypes. As I mentioned earlier, BRCA1 mutations significantly increase the predisposition of patients to breast, ovarian, prostate, and several other types of cancers. And currently in the clinic, patients carrying these mutations often employ a number of risk-reducing strategies, which can include something like active surveillance, but often involves life-altering surgeries together with adjuvant or combination chemoprevention therapies. And so the overarching goal of the particular project I'm talking about tonight is to identify BRCA1 mutations that might be appropriate for a next generation type therapy based on this ASO technology that I've described, in order to be taken as a prophylactic to actually recover BRCA1 function in those patients that carry these deleterious mutations. So in order to begin the work on the BRCA1, I'd like to just highlight that this has been born out of a partnership with an undergraduate research teaching lab, what we call a curriculum-based undergraduate research experience at UC Santa Cruz. And the Cures lab paradigm has really been pioneered by my colleague in MCD biology, Jeremy Sanford, who several years before the pandemic started, so now probably going on about 5 years or so, has been running what he calls the RNA Cure Lab. And the main objective of the RNA Cure Lab is to have undergraduates take mutations that are known to be linked to human disease in a genetic way, and then to study those mutations in the laboratory to ask the specific question, do these mutations actually cause a problem with this process of RNA splicing that I've described? So in this representative piece of experimental data, the only one I'll show in the talk tonight, this is a gel from an undergraduate teaching lab where the students were screening different known mutations in BRCA1 that predispose patients to different types of cancers. And the main take home that I want you to sort of observe here is that there were 4 different mutations that were studied in this particular experiment. And of the 4, 2 of them, mutation 3 and 4, give a different pattern compared to the normal or the wild type in this banding type analysis on a DNA gel. And this pattern is indicative of indeed that mutation causing a problem with the RNA splicing, making these mutations rather potential candidates for ASO drug development. And so using information like this and other sources of data, of course, a number of scientists here at UC Santa Cruz have formed a consortium that we refer to as the Center for Open Access Splicing Therapeutics, or COAST for short. And the mission of the COAST is basically to identify appropriate gene targets for ASO drug development. In this case, we're focusing on BRCA1. And that can be done either through curation of human disease mutation databases, or as I mentioned earlier, through clinical partnerships with folks that we work with, for example, over at UCSF. Much of the work that we've been doing over the last several years on this project has been involved in process automation and high throughput assay technologies in order to increase the efficiency with which we can screen and discover novel ASOs. And I'll just mention as a sidebar, since Doug mentioned it, that much of the automation that we've learned to apply to this ASO discovery effort was learned during our effort to set up the COVID testing lab here at Santa Cruz in the spring of 2020. So any ASO lead drug candidates will ultimately be validated here at UC Santa Cruz through collaboration with folks who work with state-of-the-art disease in a dish tissue culture models. And importantly, I just want to mention that since the inception of COAST, we've been working closely with Jenny Reardon and her colleagues at the UCSC Science and Justice Research Center in an effort to explore some of the ethical challenges that are involved with developing precision RNA therapeutics as we're beginning on this broad objective. I will mention that the COAST consortium was fortunate to receive a $75,000 seed funding grant from the UC Santa Cruz Office of Research. And this funding, together with the money that we've received from Santa Cruz Cancer Benefit Group, has been critical in producing preliminary data, as Doug mentioned, that we're now using to apply for much larger grants to continue to push this important work forward. And so with that, I'll just conclude now by thanking the members of my group, the Stone Research Group, together with members of Jeremy Sanford's lab in the MCD biology department, who we have been working very closely together on this work. I've also listed here the founding affiliates of the COAST consortium, 5 of whom actually, as indicated by these asterisks, have been previous recipients from the Santa Cruz Cancer Benefit funding. So we're very thankful for that. And with that, I'll stop and would be happy to answer any questions you may have. Thanks so much for your attention.
Thank you, Michael. I'm going to wait for a few questions to appear in the chat, or sorry, in the Q&A. And I have one question here that maybe both of us could try and answer, Michael, which is, what is the smallest organism that can contract cancer? Would study of that organism be effective due to its greater simplicity? And that's, you want to try to answer that, Michael?
Yeah, I mean, that's an interesting question that could go in a number of different directions. I mean, the first thought that I would have is that though it might not be considered cancer per se, I would say that many organisms that are single-celled eukaryotes, like yeast that I know Doug studies in detail and many others, or for example, ciliates like paramecium that grow in ponds and things like this. So those cells have developed are interesting model systems because they require an immortal phenotype because they're just one cell as an organism, and therefore if they're not able to continue to grow, they die. But of course, that's not necessarily cancer. I don't know off the top of my head what the smallest organism is that would technically be, I would imagine it would be a mammalian organism unless it was modeled otherwise.
I think the prevalent model organism for cancer is mice.
Yeah.
They get cancer and are incredibly useful for understanding cancer progression, cancer metastasis, and cancer treatment. And along the lines of what Michael said, like my lab studies, baker's yeast, and it turns out that they have a version of one of the most evil oncogenes. That an oncogene is a mutagenized gene that causes cancer. They have their own version of it. It's called RAS, and my lab studies that. And so what we learn in yeast, we can apply to, like Michael said, mammalian cells.
Right.
Another question here, what are the ethical issues?
Yeah, so that's also a pretty substantial question. So some of the key ethical issues that we've been working and learning really from the science and justice research folks on relate to, especially when you get into the area of precision therapies, where a therapeutic is being designed to a very specific mutation found in a particular patient or a very small cohort. There arises the question of who should be at the front of the line, right? Which patients and which cross sections of society are getting access to some of these therapies. So that's a critical ethical issue. Cost of treatment is, of course, always front of mind, which relates to sort of the accessibility issue. And another interesting topic that we've discussed with the science and justice research folks is sort of this idea of a quality of life metric that we as biomedical scientists are learning from the ethicists about, that in order to justify the effort put forth toward developing a given therapeutic, bioethicists actually try to quantify like sort of how much impact in sort of quality of life years, qualities as they refer to them. And so there's a deep science to the bioethics of how to approach tackling particularly rare diseases, because many of these are not of particular interest to big pharmaceutical companies, although there are important efforts underway that are being in part motivated by some work at the FDA to sort of motivate rare disease research. But so there's a lot of ethical issues that one should consider when embarking on projects like this.
Thank you, Michael. There are a lot of really good questions here. We won't have time for all of them, but another one is, do the antisense RNA medications benefit from the RNA stabilizing technology that made the COVID vaccine so successful?
So not exactly, though. That question is like very much hitting a key point from a chemist perspective with respect to nucleic acid therapeutics. ASOs, these antisense oligonucleotides, are really, it's a very exciting time right now for ASOs because compared to the naturally occurring RNA and DNA molecules that occur in our cells, these ASOs are chemically modified in certain ways so that they resist being degraded by enzymes that are designed to chew up RNA and DNA inside living organisms and cells. And so that's been a major challenge for nucleic acid-based therapeutics, particularly RNA-based therapeutics, is the stability of the RNA. And what's exciting is that a lot of those challenges there are good solutions for those with certain chemical modifications that can be put into the ASOs. To my knowledge, though it's proprietary, so I might not know, but to my knowledge from my reading with respect to the mRNA vaccines that were developed for for COVID immunization, that has more to do with the way that the RNAs fold upon themselves, and in doing so, also sort of resist the sort of breakdown by enzymes and other chemical reactions in cells. So it's an important overlap issue, though the exact solution is not mapping directly.
Another question, since cancer cells can have multiple mutations and cancer cell clones can even change these mutations and evolve, how can the focus on a specific gene mutation make a difference? Yeah,
that's a great question. And so I think that brings up this idea that some mutations are what are referred to as driver mutations, and other mutations in cancers are more sort of passive or passenger mutations. And so what you'd be looking for here is to directly develop a therapeutic to act on a driver mutation. So for example, a mutation that renders a tumor suppressor gene like BRCA1 inactive is a really significant blow to the normal cell control or homeostasis systems. And so the idea would be that correcting that would sort of overcome some of the other challenges. The other point that I would make specific to BRCA1, because of its role directly in repairing DNA damage and mutations, genes like BRCA1 that have mutations give rise to what is called a mutator phenotype, meaning that you have a single mutation that then sort of amplifies into many different diverse or heterogeneous mutations. And so the premise or rationale of the therapy approach, or the sort of prophylactic approach that we're imagining, would be to catch that early in a carrier and to actually rescue the normal DNA repair function in a patient before the mutator phenotype has a chance to get out of control. But that's not a simple question that can be answered just with a one-off, but that's a very good question.
Great. I'd like to get to all these questions because they're all good. I'd like to keep us on schedule though and go on to our next speaker.
Okay, thank you.
Yes, thank you, Michael. So our next speaker will be Olena Vaske, a professor of molecular cell and developmental biology, and a pioneer in using genomics to treat pediatric cancers. Olena's pioneering work has led to her being awarded the prestigious Colligan Presidential Chair of Pediatric Genomics. And with that, I will let her start her talk.
Thank you very much for that great introduction. I am very excited to be here and to tell you about my team's work on using novel genomic technologies to advance cures for pediatric cancer. Now, unfortunately, kids do get cancer as well. And the stats are that about 42 children in the US are diagnosed with cancer every day. The average age of diagnosis is about 6 years old. Cancer is slightly more common in boys compared to girls. We don't know why, but it comes out that about 1 in 300 children will get cancer before they turn 20. And cancer is #1 cause of death by disease in the US and in other developed countries as well. Now, when people talk about cancer in children, they often say that this is a medical success story. We like to point out that this is just the beginning of a medical success story, and we did make some progress in this field. So if you look in 1950s, virtually everybody with a childhood cancer diagnosis would not survive. They would die from their disease. And looking now in the 2000s, virtually everybody survives for at least 5 years from diagnosis. So about 80% of patients or more survive for about 5 years from diagnosis. So great difference. Why do we say it's the beginning of a medical success story? Well, if we look long term, so if we look for 30 years from the diagnosis, only about 20% of children who had cancer would live a normal life and not have any chronic consequences to their health as a result of the cancer. And then about a quarter of children, they will survive for 30 years, but they will have some chronic medical conditions. And then another, so about 19% will have life-threatening conditions as a result of their treatment, including secondary cancers, including heart disease, infertility, and so on. And then about 34% of patients will actually die within that 30 years from diagnosis. So we still have a long way to go, and we still have progress to make. So my group uses cancer genomic approaches to develop novel therapies or to identify novel treatment avenues for pediatric cancers. And here is a brief introduction to what cancer genomics is. So we've talked about in Michael's presentation about how cancer is a genetic disease, and some of you have asked really great questions about this. So we know that every cell in our body has DNA, which is a sequence of letters A, T, C, and G, so a genetic code. And so that genetic code determines how that cell looks like, how it operates, what it does. And then you can have a spelling mistake or a mutation in that DNA. And as we've talked in the previous talk, you can have several mutations. And so these mutations alter this genetic code and result in abnormal cell growth, proliferation, which is cancer. And so cancer genomics is the study really of how mistakes in our genome or mutations contribute to cancer development. So in the past few decades, we have this sequencing technology called Illumina sequencing, which can cost effectively read the DNA of cancer cells and read the DNA of normal cells. And so this is a paradigm that's used in cancer genomics, where you have a cancer patient, and both adult and pediatric. So I study pediatric, but this is true for adult patients as well. You can take a sample from that patient, read the DNA from normal tissues not affected by cancer, often that's blood, and the tumor tissue. So read this DNA and determine what are the mutations that are found in the tumor, and use those mutations to identify therapies that could be tailored to that tumor and could kill the tumor while sparing, hopefully, the normal cells. So if you do that for children, though, in most cases, we realized you don't find a lot of mutations. So in adults, you find a lot of mutations, and we've talked about that in the previous talk. But in children, you actually have a lot of cases where you don't see a mutation, or you don't have a mutation that you can target. And in California alone, there are 400 to 500 children a year which fall in that category, where, I mentioned 80% of children survive for 5 years, and then there is 20% that don't survive. And so it's that category where we don't have treatment up front for those children that works, or those children were treated for their cancer and then the cancer came back. And so then the sequencing technology could be used to identify, to try to look for these mutations that could then help us identify therapies. But so if you do this, if you look at the DNA, in 9 out of 10 children, you actually don't find a mutation that you can target by therapy. And so those kids would still be out of treatment options. And only in one case, so this blue child, can you find the therapy. And so in my group, we want to find therapies for everybody, for all of these difficult to treat cases where we don't have a good treatment to start with, or the treatment failed. And so how do we do that? And so we hypothesized that looking at the RNA, so the molecule that Michael spent a lot of time discussing, can help us. And so I'll share with you one patient that really started us on this journey, looking at the RNA and hopefully explaining what the RNA can offer that the DNA cannot. And so this was a patient, 8-year-old boy with a rare brain cancer, rare brain sarcoma, shown here. And this was actually a Canadian patient. And so this patient was treated very aggressively, as many of the children are. That's why we have so many side effects later in life. So aggressive treatment initially controlled the tumor, but later, unfortunately, the tumor metastasized. And so at that point, there was no therapy. There was a mutation identified, this gene fusion, which I'll talk about gene fusions later, but there is no drug that can target the gene fusion. And so the data for this patient, RNA data, was shared with our group. And so we did analysis looking at the RNA. And so what we did, in addition to looking for mutations, which is spelling mistakes, we looked for abnormalities in the amount of a gene product, so amount of the RNA, amount of the transcript. And we found that this gene called JAK1 was really highly expressed in the patient compared to other cancers. And so what was significant is that this JAK1, it made sense functionally. It was downstream of this fusion, so molecular biology made sense. And what was significant is that there was a therapy called ruxolitinib that could target JAK1. And because this patient had no good treatment options, the clinical team decided to try this therapy for him. So this is a complicated graph, and I'm just showing this to you to show you real data. But essentially, Kelvin, this little boy, started responding to this therapy and responded to this therapy for a while, when before that he didn't respond to any kind of standard chemotherapy, aggressive therapy. And so this approach of looking at the RNA and looking for abnormally expressed genes as therapeutic targets, in this case, was useful. And so this really inspired us to look at RNA for other pediatric cases and really start our research effort to comprehensively look at these cases like Kelvin's. Now, what about this? So I mentioned that in Kelvin's case, and I should say that I'm using Kelvin's name because that was his wish, to have his name used in talks like this. He wanted his story known, and we really appreciate his contribution to this project. But what about this gene fusion? And so gene fusions actually are hallmarks of pediatric cancer. So about 50% of pediatric cancers have what we call these gene fusions. And so essentially you have two genes, gene A and gene B, and as a result of some molecular event, they're fused together into this chimeric product, which is abnormal. And then this product can go on to perform some abnormal functions and contribute to cancer. These are great therapeutic targets because they're not found in normal cells. They're only found in cancer cells. And so if you can develop a drug that can kill this fusion, this will be a really great thing. Now, the sequencing technology I mentioned that's used, Illumina sequencing technology, kind of the standard technology we use now, it's a short read sequencing technology, and it can detect these gene fusions, but the detection is complicated because the reads are short. And just for you to appreciate, so this is a kind of a diagram of a fusion transcript, and this is what the short reads would look like. And what I want you to just take away, there is some analytical manipulation that we need to make to derive the presence of the fusion from these short reads. So it's possible to detect the fusions, but it's complicated. What we now have is we have a new technology called long read sequencing, and there are several of such technologies, but one particular one called nanopore sequencing was co-invented at UC Santa Cruz. And so this technology is particularly suitable for the detection of fusions because you can sequence the whole transcript with one read. So here is my fusion, the short reads I mentioned, you can detect fusions, but it's complicated. And with long read, you can detect the whole fusion with the whole read. So you can just read it as a word and it will be much easier to understand, and we can understand this fusion in much more detail. And so the gift from Santa Cruz Cancer Benefits Group really allowed my lab to take this new technology co-invented at UC Santa Cruz and apply it to pediatric cancer. And one specific application is reconstructing these gene fusion events and understanding them better. So we're very excited about this. And we actually recently got another award from the UC Office of the President to support this work, to use this new technology to study gene fusions in pediatric cancer. And so in summary, so what I've told you in this talk is that childhood cancers are different from adult cancers. They have fewer mutations. And so looking at the RNA is very important. And so I've showed you some examples of how we can look at the RNA. And I've told you a little bit about these gene fusion events, which are hallmarks of childhood cancers, and how they can be detected really well with long read sequencing. And we're really, really excited about the possibility of using this technology in our lab to study these gene fusions. And so this is a team work, and I wanted to thank my team and thank all of the supporters, and thank you to you for these gifts that allow us to enter into these new areas to expand our work. Thank you.
Thank you, Olena. I see a question here. A good one is, are there common causes of childhood cancers?
other common causes.
Right.
So there has been a lot of research into the causes of childhood cancer. So one cause that Michael kind of mentioned a little bit is there are these cancer predisposition genes. So BRCA1 is not one of them, it's more adult cancer, but there are predisposition genes that predispose families to pediatric cancer. However, that's the minority of cases. It's important to know that there are predisposition genes, but there are a minority of cases. Other causes, there has been a lot of work trying to understand, is it something in utero? Is it something environmental? And there is not a good answer yet. There is some recent work that looked at de novo mutation, so a new mutation that would happen in the developing fetus that the parents did not carry, and perhaps has to do with the fact that we now have children later in life, right? Like parental age can contribute to having these new mutations, but it's unclear. So pediatric cancer affects every ethnicity, every socioeconomic group, so it affects everybody and affects everybody randomly. And so there are studies on causes, but the answer is complicated. Likely it's a combination of different things. Sorry, was that? Hopefully that helps.
Here's another question. Can gene fusion events occur in all tissue and blood cancers? Are these like chromosome translocations?
Yes, absolutely. So a lot of them occur in blood cancers like leukemia, and a famous one that some people may hear about is called the Philadelphia chromosome that occurs in chronic myeloid leukemia, actually an adult leukemia. And so that gene fusion BCR-ABL can be targeted by a therapy called imatinib, which is a medical success story. But more recently, it's been recognized that these gene fusions also happen in solid tumors like sarcomas. And so that's where a lot of childhood cancer, childhood sarcomas carry them as well. So yes, both solid and liquid tumors. And yes, as a result of translocations.
Thank you. Another question. Why are pediatric cancers treated with such aggressive treatments if they have fewer mutations than adult cancers?
Good question. I invite people, so if people are interested in that, there is a great documentary that's based on the book Emperor of All Maladies that talks about kind of the history of how combinational chemotherapy was developed for pediatric cancer. It was developed by trial and error, really. Scientists came together in the 50s and, wanted to develop some treatments because this was such a lethal disease. And they started combining chemo drugs until they found a cocktail that worked. And so because that cocktail worked, we kind of kept using it because it worked, right? Nothing else worked. But right now, research efforts focus on de-intensifying therapy because we are realizing there's so many side effects. And so now, okay, we have something that works. How can we make it less toxic? So that's
great. And I should say that we're going to try to answer some of these questions offline. We have so many good questions that we can't answer. So keep us on time. I'm going to move us to the next speaker. So our next speaker will be Sachin Parecadan, an undergraduate who is an undergraduate working in the lab of faculty advisor Shaheen Sikandar, who studies breast cancer and metastasis. Sachin received this year's Santa Cruz Benefit Group Student Research Award from the endowment that the Benefit Group created. So thank you, Sachin. You can take it away.
Hello, thank you for that introduction. And I'm happy to be here today to talk to you guys about this award, how it has allowed me to do this summer, and it has allowed me to do and the research I will be helping with over the summer. So this award recognizes our work on breast cancer and allows me to accumulate research hours to help me achieve my goal of going to medical school and becoming an oncologist. I chose oncology as a profession at a young age because when I was 7, my mom was diagnosed with stage 3 triple negative breast cancer. And when I was 12, my dad was diagnosed with large B-cell non-Hodgkin's lymphoma and follicular lymphoma. Luckily, my parents survived their cancer battles. I decided to pursue oncology when my dad's cancer resurfaced when I was in high school because I wanted to be like the people who saved both of my parents' lives. This award has allowed me to do research 6 hours per day, 5 days a week. During the past summers, I worked at places like Panda Express and an amusement park so that I could pay for rent. With this award, I can subsidize my rent while doing research full time. During research, Also leaves me time to study for the MCAT at the end of the day. The previous summer, I tried studying for the MCAT, but often couldn't because after work, I would be too exhausted to study. I can only effectively study on the days I was off. After research, however, I still have the energy to study and I've seen improvements on my practice test scores. During my spring quarter this year, I took a cancer cell biology class taught by Professor Sikandar, who's the PI I work under. One day, Shaheen brought a cancer tumor from her lab to show in class, which got me curious about her research. I read that she was studying a protein that could be responsible for breast cancer metastasis and could be a therapeutic target for treatment, including for triple negative breast cancer, which my mom had, which made me want to join her lab. I asked her if I could join, which made, and she said yes, and later on nominated me for this summer research award. In the lab, I study breast cancer metastasis. To understand metastasis, it is important to know that not all cells in a tumor are the same. There are different populations of cells that look different and have different abilities. One small population we study in the lab is called metastasis initiating cells, or MICs, which are labeled in green. These cells are able to leave the tumor and travel to other parts of the body, like the lungs, via the bloodstreams. The MICs often then get stuck in small capillaries and start growing to create new tumors. The research I am primarily helping out with is trying to identify these MICs. We have identified the protein LMO2, which is a new marker of MICs. When we look at tumors under the microscope like this, we have seen cells that express LMO2 congregating near blood vessels. When we delete LMO2 in breast cancer cells, there is a decrease in metastasis. Right now, we are generating a new system to track LMO2 cells to see if they're physically moving into the bloodstream. In the future, we can create therapeutic drugs to target LMO2 and thus reduce or stop breast cancer metastasis. I'm grateful for this, to be part of this amazing research and for this award for allowing me to be able to participate in this research over the summer. I really appreciate this generous award, and that has made my dreams significantly more attainable. Thank you.
Thank you, Sachin. I see some more great questions here. And one that is relevant to your talk, Sachin, is somebody has asked, how can students become involved in cancer research? And you gave part of the answer there. Maybe you can say a little more.
Yeah, so, well, how I joined this lab was, again, I was taking that class with Professor Sikandar, and then I went to her office hours a lot. So she got to know me before I even joined the lab. So I would say, yeah, try to read also their research beforehand, and like show you're interested in their work, and be interested in their work. And then I think that will email them saying, hey, I would like to join the lab, describe your background and why you want to join the lab. I think that will probably most people or the PIs will let you in if you show real curiosity about their work.
I'll add that that's one of the important things that we do at UC Santa Cruz. So everybody who has a biomedical research lab has undergraduates who help out in their lab. And this is how undergraduates get the research experience that they need to enter graduate school and become researchers and cancer researchers. So my lab typically has 3 to 4 undergraduates in it, and they're working as a part of a team with a graduate student or a postdoc. And it's an incredibly valuable experience for them and critical for them to get into graduate school. So. I see a few more questions here. We have a little more time. One question that came up, I think, for both Michael and Olena is, how soon do we see some of these things getting into the clinic? So for example, Michael, your work, when do you think we could start seeing that in the clinic?
Thanks, Doug. Sorry, I was just trying to deal with the bright sun, California sun that's coming into my office right now. Guess that's not anything to complain about. So, so as I mentioned, so with respect to cancer therapeutics, to my knowledge, I don't think that there are ASO drugs to treat cancers that are all the way through trials. I do know Interestingly enough, there's an interesting study on ASO therapy for BRCA1 patients, or I'm sorry, for not BRCA1, for triple negative breast cancer patients who don't carry mutations in BRCA1. And what's interesting about that is that it has sort of the inverse logic to what I described during my talk, in that the standard of care for those breast cancer patients does not work very well because there's a functional BRCA1 gene in the cancer cell. And so in that context, studies have shown that if you actually use an ASO to knock down the function of a gene, you can actually sensitize those breast cancer cells to the standard of care that are used in the clinic. And so that's an example. In other contexts, non-cancer contexts, there are a number of ASO drugs that are currently used in the clinic. As I mentioned, Spinraza as one example. There's another drug that treats a disease called Duchenne's muscular dystrophy that is also used in the clinic. And so there are several important proof of concepts. And one of the last thing I'll say about that, one of the things from a chemical perspective that is very exciting about drug delivery and the tolerability of these drugs is that because these ASO drugs are chemically effectively all the same molecule, they're all nucleic acid or RNA-based drugs, they just have different encoded specificity in the different sequences that are built into those molecules. A lot of the so-called pharmacokinetics, the way the drug is taken up and sort of distributed across the body, can be applied from other disease systems to new systems where these drugs are being applied. So the hope is that once lead compounds are discovered, that the transition to the clinic will be shorter than it would be for those first generation ASO therapeutics. And I'll stop there.
And Olena, it's clear that your work is already in the clinic. What challenges do you see in terms of bringing genomics to the clinic more?
Yeah, well, one challenge is insurance is paying for these tests. So these are new. So genomics has indeed entered clinic, and one challenge is these assays are still expensive. And can we get those reimbursed? And because these are new tests, so we have to do some work to convince, kind of authorities to have it reimbursed by insurance. So that's one challenge. As Michael was talking about equity, who receives these tests, and making sure that these tests are available equitably to all patients is another challenge right now, because these are complicated technologies, right? So they tend to be available at hospitals like UCSF or Stanford, but not at maybe smaller community hospitals. I did want to share a very exciting development for our campus. So Michael and Doug mentioned this COVID diagnostic lab that we developed during the pandemic. And so we're hoping to now leverage that experience to actually have a clinical lab on campus that would focus on genomic testing assays. And our goal with that lab is to make sure that these assays will be available to more patients. We're not attached to a specific hospital, and so that gives us some different kind of reach, and we hope to reach populations that would not otherwise benefit from these assays. And so that's exciting, and that's kind of a legacy from the pandemic for us.
And a follow-up question I see here is, why are these tests so expensive?
Yeah, well, the technology, so Illumina sequencing that I mentioned is quite an expensive technology, both in reagents, but also in labor. So you need to have a skilled technician to operate that test. And that's another exciting thing about the nanopore technology. So some of you may have heard about this MinION device, which is a handheld device that has actually traveled into space. So hopefully we can, have it be available in a community hospital. But it's a device that is much easier to operate potentially, and so maybe reduces the cost of that, having a very skilled technician operating that device. So we're really working on that as an institution, as our whole university. We're very passionate about equity and making sure that these technologies are available to patients, and not just to the wealthiest patients, but to everybody.
Great. And then another question that I saw earlier here is somebody said, I'm a math major. How can math help cancer research? And Olena, you might also be a good person to answer that question.
Yeah, absolutely. So in genomics, so we deal a lot with, we call these large data sets. So, one human genome has billions of letters of this genetic code. And so we need to sift through that genetic code to find mutations. And so a lot of these are, so we have a lot of computer scientists working with us. We have math majors helping as well. So I think in the field of genomics and bioinformatics, there is a lot of opportunities to get involved, interpreting sequencing data, dealing with large data sets. And so example problems are, for example, we sequence 1,000 of cancer patients, so let's say breast cancer, and we want to find some commonality among them because we think that the common thing is what's important and what we can target by a therapy. But Michael mentioned that there are passenger mutations. So in one particular cancer genome, we may see thousands of mutations, but only a handful of them will be meaningful. So how do we select that handful? How do we generalize across patients? How do we extract signals, from noise? And so all of those questions can benefit from your background. So I think, yes, absolutely, we would love to have more math majors involved.
And then I think a good question to end on here is a very big open-ended question that's probably on a lot of people's mind, which was contributed by somebody, and it's, why is cancer research so hard and why does it take a lot of time? And it's a great question because people have been, enormous amount of effort has been put into this over the last 5 decades, really. And in many cases, we're still at treatments that were pioneered 50 years ago. I mean, in some cases, there have been spectacular successes. Cancer is a spectrum of diseases, really. And I think that's part of the answer, is that it's enormously complex. But I'm going to let Michael and Olena give their answers to that.
Yeah, I mean, I think, thanks, Doug. I will say to the person who asked this question that this is a question that we as biomedical researchers ask ourselves together with our students all the time. I do think, as many people on the call will appreciate, that unlike certain other diseases, which have their own challenges, cancer is indeed, as Doug put it, a spectrum. It's really a collection of diseases that are caused by a wide variety of sort of insults to the normal properties of cells. I think part of the answer to the question from my perspective, and part of what motivates basic biomedical research, in addition to the important translational work that is done, is that our understanding of cell function, and certainly multicellular organism development and function, is sorely incomplete. And I think that there's this perception in this sort of post-genomic era that we now understand, how the cell works and how organisms work. And I think that the reality is that until we have a strong understanding of how normal healthy cells and tissues function, trying to understand how to revert cells that have lost the ability to control their growth properties to that healthy state will remain a major challenge. And so we should all be continuing to advocate for the most basic of research together with applied research, I would say.
Olena, do you want to add to that?
Yeah, I think what I wanted to say is, so kind of what Doug said, so cancer is not one disease, but really it's as many diseases as there are us, because cancer is a disease of our genome, and we're all different, we all have different genomes, and so all of our cancers are slightly different. So that's one challenge. And then the second challenge, it's a moving target, right? And I think somebody had a question about that. So you start out with one cancer, one collection of mutations, then you treat it, and then the cancer evolves, right? It's kind of like COVID pandemic. We can't get a handle because it keeps changing, it keeps evolving. And so cancer is the same thing. It's a moving target. It keeps changing on us. And so it's a challenge to kind of hit it so it doesn't come back and does not evolve further.
And Sachin, as a beginning cancer researcher, maybe you can comment on why it's so hard and takes so long.
Yeah, I think, yeah, because everyone, there's no one person's cancer is not the same as another person's cancer. So, there's like chemotherapy where it targets just fastly dividing cells. But then some person's body will, the cancer will like pump out the cytotoxic chemotherapy drugs, and then now that no longer works. And then some people that have cancer have X mutation, and another person has another Y mutation, and it's just there's different mutations and each person that has cancer has a unique mutation to them, most likely. And like there's not like an Advil you can take that will just cure all cancers. So it's just cancer is very unique to each person that has it. So it's been very difficult to treat it.
As anyone who's worked in a lab knows, it's long hours and hard work and lots of failure. But I'm encouraged. I'm really encouraged. I think research is moving faster than ever now. And with these new tools that we have and new assays and this critical mass of researchers, I see we're just making constant progress on cancer. And it's not like there's going to be a magic bullet. There's going to be 100 magic bullets. And we're starting to discover those magic bullets. So I think we all have good reasons to be hopeful, but still lots more work to do. So that I think is a good place to end. If anybody wants to contribute to the helping grow the endowment that supports undergraduate cancer researchers, I think the link for that is in the chat. Yes, it is. It's there. And if any of you want to support overall research, contact anybody on the panel, and we'd be happy to help you do that. But I want to thank everyone for coming to this webinar, and also want to thank all the panelists and for their amazing research.
Thanks
Thank you.
All right. Thanks, everybody.
so much.